Welcome for joining us today. My name is Tanya Murkiap. I'm Project Manager with the Alliance Coordinating Office. And on behalf of the Canadian HIV Vaccine Initiative Research and Development Alliance Coordinating Office, I'd like to welcome you to our webinar. For those audience members who aren't familiar with us here at the CHVI Research and Development Alliance Coordinating Office, or ACO, we were established by the Government of Canada and the Bill and Melinda Gates Foundation in November 2011 at the International Centre for Infectious Diseases, a not-for-profit, non-governmental organization based in Winnipeg. You can find out more about us or our partners on our website. Our webinar today, Bioinformatics, Phylogenetics for HIV101, will be presented by Dr. Art Poon, who is a senior research scientist in bioinformatics at the BC Centre for Excellence in HIV/AIDS and assistant professor of medicine at the University of Manitoba. Here in Manitoba, I apologize, at University of British Columbia. This webinar will provide Canadian researchers and members of the Canadian HIV Vaccine Initiative Research and Development Alliance with an introduction to phylogenetics for HIV, understanding the methods used in sequence alignment, reconstruction, phylogenetic clustering, as well as basic concepts and applications of phylodynamics. The webinar will be of particular interest to HIV researchers and trainees that want to learn more about the evolutionary biology of HIV or more specifically phylogenetics. But as always, everyone is welcome. Before we turn it over, I just wanted to get a quick sense for Art and who was on the line. So we have a few polling questions. So I'm going to quickly go through those as quickly as possible. We have like 10 seconds for each. So if you could quickly answer the following questions. Do you consider yourself to be an early career investigator? Yes or no? Okay, terrific. Our next question is how is your audio connected for this webinar? Are you using your computer or are you using a telephone? That's terrific. Thank you. Our third question is which pillar do you most identify with? Basic sciences, clinical sciences, epidemiological science, social science? Okay. Basic science is represented. Wow. Our fourth question. In which sector are you currently working? Academic research, community-based research, industry research, with an international organization, government, or other? Okay, last question. We've heard that there are some folks that have a group of or a boardroom, and they've got a group of people listening in. So if you have more than one person in your that are viewing, please type it in there. That's terrific. Okay. Without further ado, I want to introduce to you, it's my pleasure to introduce Dr. Art Poon. Dr. Poon is a senior research scientist in bioinformatics at the BC Centre for Excellence in HIV/AIDS and an assistant professor of medicine at the University of British Columbia. His research focus is the development and application of new methods in bioinformatics, phylogenetics, and machine learning to the study of virus evolution, with an emphasis on human RNA viruses like HIV and hepatitis C virus. He is a major contributor to the development of phylogenetic software package HiFi and has authored 60 peer-reviewed articles on virus evolution and phylogenetics. Dr. Poon is the recipient of a Scholar Award from the partnership between the Michael Smith Foundation for Health Research, the Providence Health Research, sorry, Healthcare Research Institute, and St. Paul's Hospital Foundation, as well as a New Investigator Award from the Canadian Institutes of Health Research. So, without further ado, I will turn it over to Art. Um,
Okay, Thanks very much, Tanya. First off, my input might be different from Tanya's, so please let me know if if you need me to speak louder or to reduce the input either with the chat box or yeah. Okay. So first of all, my thanks for the Alliance Coordinating Office for this opportunity to give this webinar. Obviously, this is a topic that's near and dear to my heart, and I really enjoy talking about it. So I hope that this is enjoyable for you as well. I have some questions that were contributed that I got by email that I'll address at the end of the webinar. I didn't have time to make slides for them, but I will talk about them. I did receive your questions. Okay, so I'm going to be talking about bioinformatics, the application of bioinformatics and phylogenetics for the study of HIV. And let's see, I've got to use this arrow key here. This is a quick outline of what I'm going to be talking about. First of all, I'm just going to be... Hello? Okay, I'll just, first of all, give a quick introduction about basic concepts in phylogenetics, and then the sort of most difficult and boring section of the talk I'll put in second on how you go about sequence alignment. It's kind of the sort of thing that you need to understand if you're going to build a phylogeny and understand whether you're using your data correctly. Third, I'll be talking about some methods for building trees. And then I'll go into sort of a new application of phylogenies, which is the area of phylodynamics, where we're trying to answer epidemiological and immunological questions with phylogenies. And finally, I'll talk a bit about phylogenetic clustering. Okay. Oh, good, the arrow key works. Sweet. Okay, so first of all, what is phylogenetics? So a phylogeny is a tree-based model of how different populations are related through their common ancestors. And I'd like to get a pointer here. All right, great. So this is a little drawing that Charles Darwin famously put together a long time ago. And it's considered one of the earliest examples of a phylogeny. So this is a tree with a root and branches that radiate out relating different species. And species, in the case of viruses, we consider different infections or different virus populations within individuals to be analogous to different species. So for phylogenies, the populations may be taxonomic groups like species, or for pathogens, the populations within different hosts. There are different ways of visualizing a phylogeny. So one is an unrooted rendering of a phylogeny or a tree, where we have the tips connected by branches to common ancestors. Now, this root here represents the earliest point in time, and for an unrooted phylogeny could be located anywhere along that tree. The analogy I like to use is that if you imagine this tree is flat on the table, like a bunch of strings tied together, and you go down and you reach down and pick it up at some point and then pick it up, then time flows from your hand down toward the table, and what you're picking up is the earliest point in time. Now, that is not indicated by this diagram. I just located the root arbitrarily here. In this type of visualization, though, we are asserting a hypothesis about where the root is located. So this is the same type of tree, only now the tips are now arrayed on the right side. We have this implies that time is flowing from the left to the right of the diagram. So as these branches connect the tips to their common ancestors, they eventually converge to the roots. So using phylogenies is really important for understanding HIV evolution. It's especially useful because HIV has a very high mutation rate. This mutation rate is due to the reverse transcriptase that's encoded by the virus that's converting its RNA genome into complementary DNA, and that enzyme lacks a proofreading ability, so it makes a lot of mistakes while it's copying. or performing that replication. As a result of this high mutation rate, a single HIV infection will accumulate genetic differences from the variant that was transmitted within weeks of transmission. So we have differences that are accumulating among diverging populations very rapidly among infections. This diagram, by the way, depicts the amino acid sequences of HIV envelopes, so different colors corresponds to different amino acid residues. This is the alignment of a collection of a particular subtype of HIV. I made this so long ago, I don't remember which one. But I'm trying to give you an idea of the amount of diversity there is among these infections at this level of amino acid variation. Oh no, someone can't see the slides. Tanya, is there a problem? Do you see the slides, Tanya? Hello? Okay. If anyone else can't see the slides, please let me know, and we'll try to address that on this side. Okay. Okay, Tanya, are you?
Hi there.
Yeah, we have some.
Here. It
Okay.
might just be.
We have a couple of attendees that can't.
Can you just let?
At least one.
Okay.
Okay, I guess it's Evasi who can't see them.
Okay. So I'm just gonna double check something. Can we get on the chat? Just confirm whether you have seen.
Yeah, it sounds like everyone else is okay though.
Okay. I'm just gonna go back one, and then forward one, and just make sure that everyone can see that.
Okay.
Okay. You know what? You carry on, and we'll until we hear otherwise. It might just be a bandwidth issue for Evasi, who's in Nigeria. So there might be a slight issue.
Okay, we'll do. Okay. Sorry, Evasi. All right, I'm going to push on then. So I'm just presenting a little gallery of different phylogenies of HIV here. This is a phylogeny relating different HIV and SIV, simian immunodeficiency virus isolates. This is a phylogeny built up from GAG sequences, if I remember correctly, and it's depicting the origin of HIV1 group M, which is responsible for the global pandemic, and its relation to chimpanzee isolates of SIV. And it gives you, this is to give you a bit of a sense of the scale of the variation relating these different viruses and where other HIV1 types have originated from. So we can use phylogenies to get at the deep history of this emerging virus. Zeroing in on the group M diversity, this is a figure that's illustrating the different subtypes of HIV1 group M. And I'm trying to give you some sense of how this diversity is structured by the spread of the pandemic in different regions of the world. So we have the origin of HIV here around the Democratic Republic of the Congo, and then the radiation of different variants of the virus that establish subtypes for subtype B through North America and then onto Europe, and subtype C largely in South Africa, and then subsequently over to Asia, to the East Asia and Southeast Asia. Third, if we zoom in some more and look at the diversity of a virus within a given host population, we find that the structure of the phylogeny, this left side here depicts a phylogeny relating HIV infections among individuals in a given host population, that the shape of that phylogeny somewhat mirrors the shape of a transmission tree. So this is a transmission tree that was assembled from conventional epidemiological methods, contact tracing. And so this is a diagram from Thomas Leitner's classic paper in PNAS showing how the shapes of those two trees are similar. So the virus is evolving so quickly that there is adequate information to reconstruct some of these relationships. And finally, this is a phylogeny from the classic data set published by Shankarappa and colleagues. I think this was patient 9. This is a tree relating different envelope sequences from a single host over roughly a decade. And this is a tree relating all, well, not all, influenza A virus sequences sampled from around the world. So here, just to give you an idea of the scale of the diversity that can accumulate within a single patient compared to the scale diversity for influenza around the world in a given seasonal outbreak. Okay, so hopefully with that gallery, I've given you a sense that phylogenies are useful, and there's a reason to listen to what I'm going to talk about for the next 45 minutes or so. It has many uses. We can perform comparative studies of genetic variation. It tests hypotheses about evolution, reconstructs times since common ancestors, and the way that's been done, for example, is trying to estimate the date of the origin of the HIV pandemic and reconstruct ancestral states. And one application of that is trying to reconstruct what the variant that was transmitted to establish a new infection looked like. What was the sequence of that variant, given that it's almost impossible to sample it directly. Okay, so the first thing that we really need to do, assuming that we have a bunch of sequences, is that we have to know how those sequences are related to one another. An alignment is a hypothesis about which bases in two or more sequences were descended from the same ancestral base. So there's some parent sequence that is the ancestor of two sequences that we're looking at, and a given base, we want to know if those bases were copies of the same base in the parent. Now, this would be fairly straightforward, even with some mutations, except that there's insertion and deletion. And in some regions of the HIV genome, there's boatloads of insertion and deletion. So this becomes a difficult task. But this is something that we have to do if we want to even think about making a tree. So in order to perform an alignment, we have to make some general assumptions about how those sequences have been evolving. And one way of doing this is to postulate what's known as a score matrix or a substitution matrix, which makes a set of general assumptions about how often one particular character, that is like a nucleotide or an amino acid, how often that character is replaced by some other character. This diagram depicts one of the point-accepted mutation matrices, or the PAM matrices. This was curated by Margaret Dayhoff, arguably the founder of the field of bioinformatics. And I've colored-coded it so that green indicates that these amino acid substitutions happen more often than some baseline levels. So this would be aspartic acid being replaced by glutamic acid, I believe. And that would be something that would happen more often than, say, that residue being replaced with cysteine. In addition to score matrices, where we're trying to develop some assumptions about the way that evolution happens, we also need to make some assumptions about insertions and deletions. And in order to accommodate those, we have to use what's called gap penalties. A gap represents a base that is absent in one of two sequences if we're doing a pairwise alignment. So there's some base in the other sequence that has no homolog. That has no base in the second sequence that it is related to. By convention, we represent these cases with a dash. The missing base is represented by a dash. And we refer to them as indels, which is a sort of a portmanteau of insertion deletion. If we don't know what the ancestral state is, we don't know if it was an insertion along one lineage or a deletion along another lineage that led to this case where we have a mismatched base between two sequences. Now, we have to, when we're performing an alignment, we have to penalize an alignment for these gaps. Otherwise, our computer is going to happily allow the sequences to slip along and have no matches on the other side and give that some arbitrary score. So here's a sort of a simple example of how these scores and gap penalties would work. We would assign any matching nucleotides between sequences with a score of +5. Any transversion differences would get a -4. Transitions would get a -2 because they happen more often than transversions. And we'll use a gap penalty of -10. So if we're starting with the sequences that we had in the previous slides, if we had this alignment, then we would add 5 and 5 and 5, for those are matches, a gap penalty of -10, transversion of -4, so we can follow along here, and finally a match. And so I'm just summing those together and driving a score of 6. And here is a slightly different alignment where I've moved the T over here, and we add up 5 plus 5 plus 5, a transition penalty, which is -2, and a gap, and finally a +5, and that's all here. We get a score of 8, and so this is a slightly better alignment based on our assumptions about how evolution works. So what if you know that one of your sequences is only a partial sequence? In that previous example, we kind of we could assume that those sequences are basically going to align end to end. So they're both covering a given section of the genome completely, notwithstanding some insertions in the middle or deletions in the middle. If we wanted to align a partial sequence that we knew was incomplete against a complete sequence that's much longer, this is going to require a lot of gap penalties, and we can end up with a situation where one of these sequences is scattered across the length of the other. Now, this is fairly unlikely, and so what we need to do is use the information that, okay, we know that we didn't get the entire sequence, and so we know that there are going to be some gaps on either end of the alignment, and so we're going to allow the program to ignore these gaps at either end of the sequence. That allows it to keep these bases together. And these would be known as terminal gaps or prefix and suffix gaps. So I've just quickly gone over some of the basic concepts of pairwise alignment, where we have two sequences and we want to figure out how the bases that comprise those sequences are related to each other. That's useful, but it's a lot more useful to be able to have an alignment of many sequences and how all of those bases are related together, so that we can read down a column of the alignment and see a bunch of bases that look more or less the same. It's not straightforward to apply our tool of pairwise alignment to generate a multiple sequence alignment, and this is the reason why. Suppose that we have 3 sequences, so this is sequence 1. This is, oh wait, so I have to drag this guy here. This is sequence 1, this is sequence 2, this is sequence 3, and sequence 3 is longer. So suppose that we're doing pairwise alignment against sequence 1. If we do these alignments, then we're going to have good pairwise alignments of 2 to 1 and 3 to 1, but we can't stack those, like we can't stack sequence 3 here and expect to have a good multiple sequence alignment, because 2 hasn't been aligned to 3. And that gets worse as we add more sequences to the data. So we have to have some procedure for applying our tool of pairwise alignment in a way that we're going to eventually produce a multiple sequence alignment. The way this is done is that we sort the sequences into a guide tree that is going to determine the order that we gather pairs of sequences and align those together. So in this guide tree, we populate the tips of that guide tree with the unaligned sequences and then start aligning pairs of sequences from the tips of that guide tree down to the root. So this is sort of counter to what I had said from the outset, that the initial step of a multiple sequence alignment usually involve making a tree, but I just said that we have to have an alignment in order to make a tree. I wasn't lying. Well, the way that this is done is that we have to come up with some alignment-free method of generating a guide tree, and that goes into topics that I won't touch on for time considerations. But there are some alignment-free methods for generating a sort of a rough and ready tree that is good for alignment. And those methods tend to distinguish different alignment packages out there, like Muscle uses a Kamer approach to generating a guide tree. By the way, if you want to, feel free to interrupt with questions in the chat window if something comes up, and if it's going to be fairly brief. Otherwise, I'd be happy to take more comprehensive questions at the end. Okay. So, multiple sequence alignment it's computationally complex, which is a fancy way of saying that it's going to burn a lot of processing time on your poor little desktop computer. This is especially true if you're working with thousands of long sequences, and in HIV we're collecting many, many sequences. It's not unusual for us to work with 30,000 sequences and up, and those will be fairly long. And so multiple sequence alignment, as I've described it, is basically not an option. We can't wait for days for the computer to try burning through the size of data set. So something that I've been using is pairwise alignment to a reference, which I just said is not a good idea. But there's a way of generating a sort of a quick, rough and ready multiple sequence alignment using pairwise alignment to a reference. And that is discarding insertions relative to that reference. So I'm going to go back to this example here where we have sequences 1, 2, and 3. And recall that this sequence 3 is longer than sequences 1 or 2. It's got this extra C in it. If we enforce this rule that any base that is an insertion relative to the reference, that it causes the reference to have a gap appear in it, we're going to throw that out. And so this C here gets clipped out. And so this whole column here, colored in red, gets popped out. These collapse in, and now those two sequences are the same length. In fact, they're identical, and we can stack them onto this pairwise alignment. Now, this has a couple of advantages. One, it's really fast. And second, it enforces the coordinate system of the reference. What I mean is that you can, if you've aligned everything to a given reference, like let's say HXB2 pol, and you can read along that sequence and look at this C and say, oh, this is C243 of HIV HXB2 pol. And everything down from there would be in that coordinate system. So now it makes it easier to interpret positions in your alignment. The downside is that this is not going to be as accurate as full-on multiple sequence alignment. Okay, so just to wrap up this section, there's no one method that you can use that's going to guarantee that you're going to get a correct alignment. You can't. There's not a likelihood function that you can maximize to guarantee that you have the correct alignments. All of these methods that I've described, they're arbitrary or heuristic methods that give us an alignment that we hope is pretty good. There are methods out there, incidentally, that do use Bayesian methods to sample alignments from the posterior, but I've only seen those applied to alignments of 10 sequences, and it becomes really intractable to look at larger data sets. They're kind of neat models, though. One other caveat, you should always look at the resulting sequence alignment. So if you stuff your data into alignments the software package on the web or on your computer, it's going to produce another file, and you could happily toss that file into another program that's going to make a tree. But you really, really should open up that file in some visual alignment editor like AliView or CL used to be an option for the Mac. PC folks have something else that I don't remember right now because I don't use Windows, sorry. but I'm sure that there's, yeah, what is it, SeqEdit? I think SeqEdit is one of them. Yeah, thanks, Sabrina. Bioedit is one. Okay. Okay, when you're writing up your methods in a paper, the right thing to do, the due diligence, is to give the specific methods that you use to generate the alignment. That includes the substitution of the score matrix, the gap penalties, whether or not you use terminal gap penalties or not. That is in the spirit of making your science reproducible. We seldom do it, but it's something that we should be doing more often. And finally, this is an HIV-specific point. There are some regions of HIV that you just will not be able to align. And that's because there is so much insertion and deletion that there's no sense of sequence homology. So you can't look at, for example, a region of V1 and V2 and say, that string of nucleotides is related to some other string of nucleotides in another sequence. Because that string of nucleotides came from some mystery place because it's an insertion relative to the other sequences, and there's no sense of it being related to anything else. We don't know where that the bases that comprise that insertion came from. That's sort of a big mystery in HIV phylogenetics. So I advocate actually clipping those sections out, at least when you're trying to reconstruct the phylogeny, because it's not going to play along with the assumptions that we have to make in order to align the sequences and make a phylogeny. Now, it would be okay to then throw those sequences back in after we have a phylogeny and then test some evolutionary hypotheses about how those insertions are evolving or how they're influencing some phenotype. But it's not. It's really difficult to try to use those sequences for phylogenetic reconstruction. Okay. So I'm going to pause for a sec there and see if anyone has any questions on that last section. I see that some people are typing. If it's a quick question about section 1, sure. Region slide 9, transmission tree. Okay, so yeah, I can do that. Let's zip back to. Sorry if I'm chewing up your bandwidth with skipping over slides. All right, there's a question about how this phylogeny recapitulates the transmission tree. So if we look at, for example, here in this phylogeny, sequences from patient 2 and 3 are more closely related than other sequences in this data set. And in this transmission tree that is sort of a visualization of a contact network gathered with non-genetic methods, we see that patients 2 and 3 share some contact. And so that's an example of how the phylogeny is in some way congruent to the transmission tree. It's not going to be the same, and we shouldn't use it to infer specific transmissions. I'll get into that later, but that's generally how those trees are the same. And so here again, here's patients 8 and 9, and we have sequences 8 and 9 that are fairly close in this transmission tree. Okay, and there was a comment about regions of interest with evolutionary information in HIV envelope. So I don't quite understand that question. I would say that the variable loop regions, the regions that I just suggested that you should clip out of the alignment before making a phylogeny, those are, yes, those are, of course, really interesting regions, but in my view, you can't use those for making a phylogeny because they're generally really difficult to align. If you're looking at within host variation, then it's fairly doable, but if you're trying to do something among hosts and you're looking at a lot of sequences, it can get to be more trouble than it's worth, and it could make your phylogeny actually less accurate. Having said that, once you've built a phylogeny from the alignment where you've clipped out those those regions, I think that you can use comparative methods to analyze those variable regions using the phylogeny after having built the tree. Okay, and if there's anything else on that, I'd be happy to take it after I get to the slides. Thanks very much. So this section is going to give a quick summary about how we build trees. And there's generally 3 classes of methods. There are others, but the ones that are used most often in phylogenetics, first of all, there's maximum parsimony. Actually, I'm lying. No one really uses maximum parsimony anymore, but I feel compelled to mention it. The problem with so first of all, maximum parsimony is a kind of a minimum evolution method where you're trying to find the tree that requires the fewest evolutionary changes to explain the data. The problem with maximum parsimony is statistically inconsistent. What that means is that it's, in some cases, can be guaranteed to give you the wrong answer even if you add more data. So that's not good. The second class of methods are distance-based methods. Distance-based methods are fast, and that makes them good for dealing with really large data sets. However, they're not necessarily as accurate as likelihood-based methods. And I'm going to go into what I mean by each of these in subsequent slides. The last type are likelihood-based methods. They're a lot more complex and hence slower to compute than distance-based methods, but they can be more accurate. So I'm going to be talking about distance-based and likelihood methods in this subsequent slides. For distance-based methods, you need to have a distance. And so we need some function, that's a genetic distance, that quantifies a distance between 2 genetic sequences. Now, the most intuitive genetic distance would be the Hamming distance. And that is basically just counting the number of bases that are different between 2 sequences. Right? That's very simple, easy to understand. It's also lousy. So one of the reasons why Hamming distance is lousy is because it doesn't account for multiple hits. And so you're going to underestimate how much evolution there's been. Actually, I'm sorry, you're going to overestimate the amount of evolution there's been based on, no, sorry. I shouldn't be doubting myself here. Yeah, you're going to underestimate the extent of evolution. And so I'm going to just work through this example here. So this is an example of how we're going to apply Hamming distance between these 2 sequences. We have one difference here, and so we have a Hamming distance of 1. And there's been an A to G substitution. Now, suppose that there has been 2 substitutions at the same site. So we've had a change from A to a G and then to a C. And so that, let's say that occurred all along the lineage leading to sequence 2. We would still give us a Hamming distance of 1, even though there's been roughly twice the amount of evolution. Finally, if we had a A to G substitution followed by a back mutation or reversion to A in this lineage, there's been a bunch of evolution, but we have a Hamming distance of 0. Okay, so in this case, our Hamming distance is underestimating the amount of evolution that's taken place. There is a family of genetic distances that are much better that account for multiple hits. You're going to see these acronyms pop up quite a bit if you're wandering around through phylogenetic websites or software. I just wanted to go over some of these acronyms so that it gives you some idea of what they mean when you encounter them. Chooks-Cantor is one of the first genetic distances that accounted for multiple hits accumulating between sequences. It made Jukes and Kenner made a lot of very strong assumptions, like that the base frequencies were equal, everything is present, A's, C's, G's, and T's are present at 25%, that substitution rates were equal, there was no transition transversion bias, but they accounted for multiple hits, and that is a valuable addition to the distance. And so this is known as JC69. Felsenstein 81 allows for unequal base frequencies, so that's relaxing one of the strong assumptions of Jukes-Kenner. HKY85 stands for Hasegawa-Kishino-Yano, and that allows for, now we're relaxing the assumption of equal substitution rates. Now we can have transitions occurring more often than transversions in the genetic distance. TN93 is Tomorane, 1993. This is one of the favored genetic distances for HIV because of the hypermutation you tend to get a more AG, G to A transitions than you do involving CNT. And so if I'm doing a neighbor joining tree with HIV sequences and I'm dealing with a lot of data, I usually use TN93. And finally, if you've got boatloads of data and time, then you can fit the GTR model, the generalized time reversible model. That means that you have 6 rate parameters, 6 substitution rate parameters, but you assume that they're symmetric. And so the rate of going from A to G is the same as going from G to A, but that is distinct from all the other 5 symmetric rates, like A to C, C to A to T, and T to A, and so on. So given that we have genetic distance, we can calculate a pairwise genetic distance matrix for a set of sequences. And that matrix is the substrate for employing neighbor joining, which is probably the best distance-based method out there. The general idea behind neighbor joining is that we're going to start with a star phylogeny where we haven't resolved any of the ancestral relationships among these sequences. We have 5 sequences, A through E, and we have some star tree relating them. Then we're going to look for the pair of sequences that have the shortest genetic distance. In this case, it's A and E. And then we create a new node in the tree that we can call AE. And then calculate the pairwise genetic distances of this new node to the remaining sequences. Okay. Then we repeat. We have another star tree relating B, C, D, and AE, and then we want to find the shortest distance relating any of those 4, any pair of those 4 sequences. In this toy example, it's AE and D, and we create a new node here, and then we would recalculate those pairwise genetic distances. And we keep doing that until we've resolved the ancestral relationships in that tree. Okay? And that turns out to work really well at recovering the topology or the order of branching events in your tree. It's not as good at recovering the branch lengths. And the reason is that we're relying on the comparison of 2 sequences to figure out the length of a branch. In this method I'm going to go through, we're using all of the data to calibrate some model of evolution, and so we're using all of the data to estimate lengths of branches. So in maximum likelihood, we have to have some model of sequence evolution that we can use to calculate the likelihood, or it's kind of like a probability, for a given tree given the data. So the objective is to find the tree that was the most likely to have produced the sequences that we've observed for a given model of evolution. And I'm illustrating this here with 3 different trees and just some arbitrary likelihood scores associated with them. And so this would be the tree that maximizes likelihood and would be our estimate of the true tree. The difficulty of this method is that there is a massive number of trees that we would have to explore if we want to do this exhaustively and compute likelihood for every possible tree. That's not going to be feasible. So we have to come up with some heuristic method, some sort of arbitrary set of rules that we hope is going to give us a pretty good answer of maximum likelihood estimate for the tree. So this method is slow, but it's more accurate than using neighbor joining. Finally, I'm going to touch on Bayesian inference. So Bayesian inference also relies on likelihood, but whereas maximum likelihood is attempting to find one tree as our best estimate of the underlying tree, with the Bayesian method, we're trying to sample trees at random from what's known as a posterior distribution. Essentially, we're trying to sample trees that have a fairly high likelihood. Now, that's not entirely true. The posterior distribution is a product of the likelihood and our prior distribution, which represents our belief about what the tree looks like before we look at the data. I don't have time to go into the technical details of that, but that hopefully gives you the general sense of what's going on with Bayesian inference. And what that does is that gives us a bunch of trees that gives us a better representation of what we know about the shape of the tree given the data. A better representation than if we relied on just a single tree. Using Bayesian inference generally is limited to trees that aren't too large. The sort of rule of thumb that I've seen on some of the BEAST boards is used to be fewer than 200. I think it's increased to fewer than 300, and then depends on when those tips were sampled. But it would probably not be a good idea to look at several thousand sequences, for example. If you were going to do that, something that the cool kids are doing with BEAST, and BEAST is a software package for doing Bayesian inference with trees, is to fix a single maximum likelihood tree, you can use neighbor joining tree, and then you turn off the proposal functions. So there are functions that are trying to change the parameters of the model. You turn those off so that BEAST has to use a particular tree shape, and then you can slide the branch points of that tree up and down, but you can't change the branching pattern. If you were going to look at a very large data set, that's the thing to do these days. And I'd be happy to go into the details of that with individual people later on by some mechanism. I mentioned that there's a large number of trees with larger amounts of data. This is just to give you a sense of what we're dealing with. If we have 10 tips, if we have 10 sequences and we're trying to find a tree, there are about 30 million different trees, and so that's like the number of miles to Pluto, just for 10 sequences. With 20 sequences, we have 8 times 10 to 21 possible trees, which I figured was roughly the number of atoms in a gram of pure tin. And that's not really intuitive, but I think it's kind of fun to think about. If you look at 52 different sequences, now you're starting to get around the number of atoms in the universe as the possible number of trees. And then we could just forget about with trying to figure out an analogy for the number of trees relating 100 tips. There's a large number. Just quickly, and I think this slide would be available for people later on, so I won't go into it in too much detail. These are some software packages for applying these various methods that I've mentioned, and I've provided URLs as well. Okay, so I'm going to move on to a section on phylogenetics. So I've gone over some methods for processing sequence data, getting an alignment, using that alignment to reconstruct a tree, and then we could stop there and be happy with, oh, these are how my sequences are related. But there's a lot more that we can do with this tree that we've just built. And this is because the shape of the tree can tell us something about how the epidemiology and immunological processes that have shaped that diversity. That concept is sort of the foundation of phylogenetics. That is that the shape of a virus phylogeny, at least for viral phylogenetics, the shape of that tree is shaped by the epidemiological and immunological history of the virus. And so This is a new field, a fairly new field. We're trying to develop methods for reconstructing these processes, these epidemiological processes, for example, from tree shapes. And this generally is done by fitting some model of epidemiology to the shape of the tree. Now, what do I mean by the shape of the tree? These are some figures that I popped out of Brian Grenfell's paper in Science, showing you different virus phylogenies. So here's one of measles, there's influenza A virus, dengue, the dengue relating different serotypes of dengue, or genotypes, and HIV, hepatitis C virus, and HIV within a host. This is probably also a Shankarappa data set. And so we have a large diversity of tree shapes here. Okay, and so that first tips people off that why do these trees have different shapes? It may be because these viruses are experiencing different environments, and that's determining the shape of the tree. So in order to talk about tree shapes, we recognize that there's variation in tree shape, but we need to be able to measure that. In order to measure tree shape, we have to break it down into two parts. There's the topology and then the branch length. So the topology is the order that lineages converge to common ancestors. It's the branching order. There's no concept of lengths involved here. There's just connections between tips and their ancestral nodes in the tree. The branch length represents how much evolution has taken place, generally, how much evolution has taken place between the ancestor and the descendant on the given branch. So to illustrate, here's a tree, and then here's a tree with a different topology, but the same branch lengths. Okay? And then here's a tree with the same topology, same branching order, but different branch lengths. So this is a sort of illustration of these different components of tree shape. So one of the ways of looking at tree shape is looking at the accumulation of lineages over time. What I mean by lineages is the number of horizontal lines. So if we take a slice here, that we would have 4 different lineages at that point in time. And we can plot that out. So this bottom axis represents time, this y-axis represents the number of lineages. And so these two trees have different lineage through time plots, or different trends. So we can then, this sort of extracts a piece of information of tree shape that we can use to test hypotheses or to fit a model to. And this originated from evolutionary biology and study of speciation from Sean Mee and colleagues. A very similar way of looking at tree shape is coalescence times. Coalescence times are basically like branching times in reverse. So a coalescence happens when two lineages, we follow two lineages back in time, and they converge to a common ancestor. That's a coalescent event. Kingdom's coalescent. I don't think I should go into that for time considerations. But the idea of coalescence stems from this population genetic model that is describing it defines our expectation of when coalescent events are going to happen going back in time. So when you look at phylogenetics in the literature, the vast majority of those papers are using coalescence times. And this is this is the branch length component of tree shapes. And that's largely because people like to use BEAST. And that's fine. But it's become a very dominant software package in the field. And so there's now interest in developing other methods. Now, I just mentioned that BEAST is using branch length, and that there's another component of tree shape, which is topology. How do we go about measuring tree topologies? One of those ways is tree imbalance statistics. So these are just two imbalance statistics that kind of measure how asymmetric a tree is. So this is Seconds index, and it counts the number of nodes from each tip to the root. So if we take this tip here, we count 1, 2, 3 nodes on the way to the root, right? And so Seconds index would be the summation of that number across the tips. Collins's index compares the number of tips to the left and the right of a given internal node. So for this node, the Collis index is a function of 3 and 2 for the number of tips on the left and right of that node. All right, so these are measures of the asymmetry or imbalance of a tree, and that gives you some information about the topology. So that is just one simple way of measuring the differences in tree shape with respect to their topologies. It's difficult to measure topology, but it's an important aspect of tree shape. And I mentioned before that software packages like Beast can apply Bayesian methods because we can calculate the exact likelihood of a model like the coalescence based on the branching or coalescence times, but we're not using that topology information. So these methods are powerful, but it's getting away from the original idea of phylogenetics, where we're talking about the entire shape of a tree. So what do we do if we want to fit more complex and realistic models to the data? If we want to relax some assumptions, and one way of addressing this problem is that we want to use as much information as we can pull out of tree shapes rather than collapsing them down to branching times or coalescence times. I just wanted to mention that this field, approximate Bayesian computation, but I see that I'm kind of running a bit long in time, so I'm going to skip over this. So I apologize for that. I wanted to mention because it's my current area of research, but in the interest of keeping this 101, I'm going to skip that over. I do want to say that this is basically a new method we're developing of using tree shapes to fit models, and this is a preliminary result where we're saying, okay, let's say that we're going to model the social network of a host population and how that's shaped the virus phylogeny. We can look at the shape of the virus phylogeny and use that to try to infer something about that social network of the hosts. So here are two different host networks that have different shapes, and those shapes are controlled by a parameter. That parameter is known as the attachment power. And so we simulated social networks and then simulated epidemics through those networks and then looked at the phylogeny of that epidemic. Using this method that we're developing, we found that we're able to, these boxes are estimates, we're able to recover that parameter of the network from the virus phylogeny pretty good. So this is upcoming stuff. I wanted to mention it, but that's all that I can say about it for this time. I wanted to get to this last section on phylogenetic clustering, so I'm skipping that piece there. Phylogenetic clustering, I'm sure you've probably heard of it. It's becoming really popular. Clustering is a type of phylogenetics because we're using the shape of the tree to extract some information about epidemiology. So a cluster is a collection of closely related tips in the phylogeny. I've illustrated this here. Here is a collection of tips related with short branches, and we're going to call that collection of tips a cluster. And we can translate that into a network diagram if we have each of these circles in the network, nodes in the network, corresponding to tips in this tree, and we draw a line between tips that are closely related, then we're converting that part of the tree into a network. And this is often referred to as a cluster as well, which is confusing. One of the issues of clustering methods, and this is generally true of all clustering methods, is that they're generally subjective. You have to make some arbitrary decisions about some criteria in order to resolve clusters from your data. Clustering is really useful. So in the context of HIV, for example, a phylogenetic cluster can correspond to a rapid series of transmission events in the recent past. And the reason is that we're looking at a bunch of sequences, and they retain a high degree of genetic similarity. And that tells us that there hasn't been that much time since those infections were transmitted from one host to another. There hasn't been enough time for them to diverge a lot in sequence. And so we're looking at a bunch of transmissions that have happened sometime in the recent past. These clusters then kind of carve out parts of this phylogeny, parts of the tree, and extract groups where, or parts of the population where the transmission rate of the virus has been high. And then we can use that information to examine factors like demographic or risk factors, epidemiological factors that are at least associated with this variation in transmission rates. Incidentally, this is a cluster that I constructed using some data from BC. Each point represents an individual. And I forget what the red ones represent. I think it was drug resistance, transmitted drug resistance. Sorry, it was a while since I built that. Okay, so there are a number of caveats to using clustering. One, that a cluster or a network a network that's built up from a cluster, it is not the same as a transmission network. So you should not look at this diagram and say, this person transmitted the virus to this person. You can't do that. This is not what this diagram is telling you. All it's telling you is that those infections are genetically similar. And we have no idea whether there's been a transmission among those individuals or not. There's a number of reasons why that's the case. Okay, I see there's a question of how I came up with that network diagram. I can address that briefly. So I'm using, actually, if I can address that question later on, I will get to it. It's a little complex. Sorry about that. So the reasons why we can't interpret a cluster as a transmission network, if you consider two sequences here, and these are sequences that are related by a common ancestor of the HIV population. This is an HIV ancestor. This is not the same as a transmission event. The transmission event could have happened at any point along these branches, anywhere along those branches. Okay, so first of all, the branching points in the phylogeny are not the same as transmission events. Second, there can be other transmission events through unobserved hosts. So this is just one possibility. Again, we have two sequences. This represents the virus lineage being in this first host, and then we have two transmission events, one through this unobserved host along this branch. Okay, so that's another example of how there's this discordance between the phylogeny, the information in the phylogeny, and transmission events. So we can't use the phylogeny to establish directionality of transmission, not without additional information. It's not the kind of thing that I think should be done. I'm certainly not attempting to do that. All that we're trying to do is use a tree to characterize variation in transmission rates in populations. Okay, so hopefully yeah, those are some caveats that I just wanted to mention, clustering. There are different methods for clustering. One is subtree clustering. There are basically two classes of methods. One is where you define clusters as from subtrees. And a subtree is a part of the tree that contains all of the descendants of an ancestor. Okay, let me flip back a bit. So we could take this ancestor, the subtree would be all of these branches and tips below it. Okay. Now, subtrees can be classified as clusters using typically one or two of the following criteria. One would be a summary statistic of branch length. So we could look at the mean length of branches within a given subtree. And what we're trying to look for is for the mean and the median length to be fairly short. So we have some arbitrary cutoff. And if it's below that cutoff, then we are inclined to interpret that subtree as a cluster. The second is bootstrap support. And the bootstrap support is our confidence in the composition of that subtree. Our confidence that the members of that subtree really are the only members that should be the descendants of a given ancestor. And I wanted to go through what we mean by bootstrapping. We have bootstrapping because it's difficult to replicate a phylogeny. So if we had a phylogeny and we wanted to make replicates, that would mean that we'd have to go back out and collect a whole bunch of independently evolving sequences from the same individuals or populations. And that would be a lot of work. So we're lazy. But we still want to get some idea of how confident we are in the shape of our tree. Okay? So what we do is that we pretend that our data is now the universe, and we go out and we do experiments in our data universe. What I mean is that we take our data set, this alignment, and we sample columns from our alignments with replacement at random. Okay, so we pull this one, this becomes the first column of a new alignment. So we're doing this to create new alignments. We grab this one, that goes in second, and this one becomes third, and we just do that until we generate an alignment with the same dimensions, same number of sequences, same number of columns. And we do that a bunch of times, and then build trees from each of these fake alignments. Now it turns out that that is a valid approach to generating replicates in the sense of bootstrapping. We build trees, and then we look at those trees, and we ask, how often do we see the same subtree in these bootstrap replicate trees? The proportion of times we see a given subtree that is a specific set of sequences related by a common ancestor to the exclusion of others, that is our bootstrap confidence in that subtree. Okay, so as a clustering criterion for the subtree, we're looking for a certain bootstrap value that's a measure of confidence of that subtree containing those particular individuals. The second method for generating a cluster is pairwise clustering. And this is where we're taking pairs of individuals and asking, do those individuals have sequences that are within some minimum distance of each other? And there are different ways of evaluating that distance. One is by calculating a genetic distance, and I went through genetic distances when I was talking about neighbor joining. We use the same ones here. Another is what's known as patristic distance or tip-to-tip distance. This is a method that I rather like. This is where we take, let me flip back to a tree here. So to calculate a tip-to-tip distance, let's say from this guy to this guy, we calculate the length of the path going through the tree until we get to that second tip. And that gives us our patristic distance. And so we're generating, we're generating clusters from these patristic distances from pairs of individuals. One of the nice things about this method is that the cluster now doesn't have to have all the descendants of a given ancestor in the tree. So if we have a scenario where we have a sort of a bunch of closely related sequences, but then one tip that's way out there on a long branch, we don't necessarily want to include that sequence in our cluster. Okay, so that's all I'm going to say about phylogenetic clustering. I've gone over sort of the diversity of methods, the two classes of methods that are used in phylogenetic clustering. There are other methods, but those are the sort of the most frequent and simplest ones. I do want to say that when you're making clusters, we have to make a bunch of decisions about our clustering criteria, and we have to vary those criteria in order to see how sensitive our results are to those criteria. Okay, that's something that is due diligence when we're publishing clustering results. Second, clustering is often done with customized scripts. What I mean by scripts like bioinformatics scripts, little programs. There are now some software packages that are being published that will allow people to do this by pressing a button, generally, what I call off-the-shelf software. This is just a sort of quick summary of some of these software packages. I've never tried any of these. I've never used them. I have no idea if they're any good, but I feel compelled to mention that they're available. Okay, and that's my last slide. I have funding sources here, and this is what I'm told I have to show at the end of our presentation. So thanks very much. I apologize if that was longer than it should have been. I do want to get at the questions that I was sent by email. So I'm just going to do that before I get to the questions that came up during the presentation. So the first question here is, what computational...
Hey,
Hello?
Art, it's
Yeah.
Tanya here. Hi, it's Tanya here. You have lots of time. You've got like 15 minutes, so take your time
Great.
with questions.
Okay. So this first question I got by email, well, first of all, something about pre-webinar reading resources, so I emailed the person with suggested readings. So hopefully you got that, and hopefully they were useful for you. The second question is, what computational tools can be used to reconstitute HIV variants from ultra-deep sequences? So what this person's getting at is what's known as haplotype reconstruction, which is a method from human genomics, where you have a bunch of short-read data, a lot of short sequences that are covering a much longer sequence. Because we have these short pieces, we don't know the linkage relationships among those sequences, and so we don't know how one short read is related to another short read that's at a different part of the genome. There are bioinformatic algorithms or software for trying to stitch together these short reads to reconstitute these long sequence variants. I really do not recommend doing this for HIV, and that's because the haplotype reconstruction methods were developed under the assumption that you have two true genomic variants, because we have two copies of our genome. And there's going to be some variation around those two variants because of sequencing error. But that means that there are two well-defined clusters in sequence space, and assigning our short reads to one of those two clusters is computationally feasible. With HIV, there can be millions of sequence variants in a given population, and with so we have millions of clusters in sequence space with some sequence error around that. It's really difficult to try to assign short reads to one of those millions of clusters. There's a great paper, I think, from Prosperi et al. In Nature Methods that tested this using a realistic artificial mixed sample of HIV clones, and they found that they evaluated a bunch of software packages for haplotype reconstruction, and it really did not work well. So I'm sorry to say you can try it, but it's not a good idea, in my opinion. Third question, use of clustering to identify recent transmission comparing IDUs and heterosexual epidemics. You will tend to find that those risk groups will sort into different clusters, and so the question then, one of the questions becomes, I suppose the frequency that that individuals appear in clusters. That's a hypothesis that you could test. One thing that you want to be aware of is whether you need to compensate for systematic differences in delay to diagnosis or time to engagement in care and the appearance of the sampling that sequence from an individual, whether that's different between those populations, like IDU and heterosexual populations. TMRCA as a tool to age the infection, how this can be applied for incidence estimation. Okay, so what this question is getting at, using the diversity of HIV sequences within an individual to extrapolate back to when the transmission occurred. And you can do that by trying to infer the root of a phylogeny relating sequences within an individual, and making use of the times at which you sampled those sequences to get it to date that root. That can be done. The confidence intervals for those estimates are pretty large, and you have to collect a lot of data in order to get at that estimate. If what you're specifically interested is in incidence estimation or reconstructing the accumulation of cases over time in a population, in the host population. Sorry, I would actually suggest using among hosts genetic variation. For example, using the lineage through time plots that I described in this presentation, that is the accumulation of branches over time, as a sort of a rough approximation of incidence. So I think that would be certainly a lot a lot easier, simpler method of getting at that kind of information than trying to estimate TMRCA for a whole bunch of individual infections. So I hope that helps for that question. I see that I have a question here about the network representation of a cluster and how that is not a network cluster transmission. So let me skip back to. Oh, I don't know if I have control of the slides. Oh, here we are. Okay, here we are. Slide 44. So, what I'm trying to get at for this is this is a cluster. This is a network diagram that is depicting a cluster that's been extracted from a phylogeny. And it's a representation of how those sequences are related. So we have lines for sequences that are adequately similar. And I'm trying to make the point that this is not a transmission network. There may be some similarities between when transmissions have occurred and the linkages in the tree, but these should not be interpreted as representing the transmission of the virus from this individual to this individual, for the reasons that I went through in slide 44. Okay, let me go back in my history a bit to see if there are other questions. Let's see, I think that I'll just get at this one being asked right now with respect to HIV envelope quasi-species. So, I think you're asking about whether we can look at sequences covering HIV envelope and using that to reconstruct lineage through time plots. To which my answer would be yes. You could, and I take it that you have, like, if you have a data set where you've already collected a whole bunch of within host sequence data, then you could probably use that to estimate the. You can probably use that to date those individual infections, given that you have sequences that have been sampled at different points in time, and that those points in time are sufficiently far apart. Okay, that would give you sort of a rough estimate of the time that the date of the infection, when that transmission occurred. Not knowing specifically the question that you want to get at, it sounds like you want to estimate incidence. The problem with estimating incidence is that you're just getting at the dates of infection for the individuals that you sampled from the population. And so that doesn't tell you anything about the other infections that have accumulated in the population. So as far as like a population level measure of incidence, I would recommend using an approach like a lineage through time plot over something based on within host variation. Okay, I hope that gets at what you're asking. Okay, see we have some more time for questions and that people are working their keyboards.
While we're waiting, I'm going to just, we can always bring this back up, but I'm just going to put the link here. Those people who need to rush off, we do have an evaluation of the webinar. But please feel free to raise your hand and you can ask your question verbally or as you have been in the chat box. But please take a moment and fill out the evaluation.
Okay, so there's a question coming up with directionality. Okay, so there are a couple of papers out there that have used estimated dates of infection to infer directionality on a phylogeny. I think there's one from UC San Diego from Joel Wortham and Susan Little and Davey Smith. It's you know it's going to be kind of shaky because estimates of dates of infection they have large confidence intervals, and so there's a fair amount of uncertainty. Even if you can label the or what we say is color the tree with respect to where the virus lineages might be in a host at a given point in time based on estimated dates of infection. First of all, you don't know whether there are other hosts in there who you haven't sampled. And second, there's a fair amount of uncertainty in those dates of infection that are difficult to accommodate in assigning those sort of host locations on a tree. I haven't seen it done well. There may be Bayesian approaches to incorporating that kind of data to infer directionality, but I haven't seen a paper out there yet that does a good job of it. There have also been papers on the topic of inferring directionality. There have also been papers on using within host variation and looking at whether a host A's diversity is nested within host B's diversity. That is to say that going back through time in host A's tree, as we go back down through host A's tree, down through the root, we have to pass through the tree of host B in order to get to the rest of the sequences of the population. That would imply that the virus lineages have passed through host B before they got to host A. Now, it doesn't say anything about whether there are other hosts on the way there. Sorry, my desktop just froze. Okay, it doesn't say anything about whether there are intermediate hosts, but that can be used to infer directionality on the phylogeny. Okay, emerging recombinants in sub-Saharan Africa. What is my view on the beast analysis for recombinants because it utilizes pure subtypes? Okay, I don't think I've read that paper. But with respect to incorporating recombinant sequences in a sort of analysis that you would carry out in BEAST, you can partition your alignments on your recombinant breakpoints. So let's say that there's a recombination event, that you know there's a recombination event in your alignment. You can look at the parts of the alignment on either side, to the left and the right of that recombination breakpoint, and then use those as independent replicates in your analysis. Okay, so then you then enforce the same assumption that there's no recombination within each of those pieces, and then carry on with your analysis. Not being familiar with this particular, I'm not sure if you're mentioning a particular study, if you're trying to refer to a particular study, but from what I can glean on your comment, that would be my approach. If there's no recombination in the alignment, and I wanted to do some phylogenetic reconstruction, for example, using Bayesian method in BEAST. Okay, see there's still a couple of minutes for questions, and I see that there's a follow-up coming on the recombination question. Okay, so the concern is that you'd lose many sequences due to recombinants. Okay. Yeah, and there's certainly, in some populations, you are going to see a high prevalence of recombinant sequences, and so what I think you're getting at is that in those cases, in some cases, it's easier to just discard a recombinant sequence from your alignment, so you don't have to split the entire alignment. With a high prevalence of recombination, you'd also have to be dealing with a lot of recombination breakpoints in your alignments. I think that one approach I might suggest is to do some simulation or look at other people's simulations where they simulate recombination and molecular evolution in sequences, and then they assess how sensitive their hypothesis test is to recombination. So there's a fair amount of papers in the literature looking at this. So how sensitive, for example, estimating DNDS metrics of selection is to recombination. Now, depending on what you're trying to get out of your data, it may be the case that the test you're trying to do is fairly robust to recombination. For example, a recombination event, a recent recombination event, it's going to tend to swap tips in the tree. It's not going to affect the deeper structure of your phylogeny. And so the test that you want to apply may be robust to a high level of recombination, in which case you may be able to justify retaining those recombinant sequences in your data. If I was reviewing a paper and it was clear that there was boatloads of recombination in the alignment, I think that looking at some of the assessment, a simulation-based assessment of how robust or sensitive the test is to that level of recombination, I think that would be really, I would be persuaded to say, okay, that's due diligence. The office have done the job, and you could do it both with and without the recombinants. Hopefully, your conclusions would be fairly robust to the removing the recombinants as well. That would be a fairly strong paper. I hope that helps. Okay.
Thanks very much. Thanks very much, Art. We're going to have to stop it there. But there was a question earlier about whether or not we can share a PDF of your presentation. So I don't know if you want to address it now or you want to just send me an email and giving us permission to share it. Then that's totally fine. But we will have the recording available in the training tab on the Alliance CC. Want to thank everyone for your participation in the webinar today, and especially you, Art, for sharing your expertise with members of the CHVI Alliance virtual community. So as I said, the webinar has been recorded. It's going to be available, and watch for announcements about its availability on the discussion forum. And take some time to complete the evaluation, please, using the link provided on your screen. Your feedback is really appreciated and helps us to enhance and improve our webinars. The ACO, along with the advice of our new and early career investigator advisory group, has developed our training program, and we're hoping, we're planning actually to have one more webinar before the end of March. We've got one scheduled for March 8th, featuring Dr. Andreas Finzi. So we hope that you'll watch for those announcements as well. So once again, thank you very much for joining us, and we look forward to your participation in our next webinar. And thanks again, Art.
Thank you very much. It was a pleasure.
Okay, bye everybody.